The embryonic period is the most crucial period, the period of organogenesis. It extends from the third week to eighth week of development. Fourth week, fifth week, sixth week, seventh week, and eighth week, the important organs and tissues are developed from the three germ cell layers, and it includes in the embryonic period is therefore considered as the crucial period of development. Third week of development, the two important structures which develop is the three germ cell layers are established in the embryo, that is the ectoderm, mesoderm, and endoderm. The second important feature which develops during the end of third week is establishment of intraembryonic and extraembryonic vascular system with the development of the tertiary villi. Fourth week of development, the embryo is about poppy seed. Eighth week of development, the embryo is about bean seed or kidney shaped. Gastrulation is therefore involved in formation of the three germ cell layers which occurs by the third week of development. The first layer to develop is the endoderm germ cell layer, which is derived from the primitive streak, which is in turn derived from the epiblastic layer. The primitive streak cells replace the hypoblastic cells and form the endoderm germ cell layer. The next germ cell layer is the formation of intraembryonic mesoderm cell. It's also derived from the primitive streak cells. The last layer, which is finally develops, is the ectoderm. Period of organogenesis therefore starts from the third week to eighth week of development, time when each of the germ cells differentiate to form various tissues and organs, which they start functioning during the between the third to eighth week. So finally coming to the important function of the trilaminar germ disc is the ectoderm is a protective in function. It forms the nervous system and skin. Endoderm forms the nutrition part of the embryonic disc. That means it forms the gastrointestinal system and respiratory system. Mesoderm is finally supposed to be a supportive system. It forms the skeletal system, muscular system, blood vascular system, and urogenital system. So end of the embryonic period, that is between the third to eighth week, the main organs of an embryonic folds are formed. As a result, the embryo, which is a flat disc, forms a cylindrical structure or cylindrical form. The external body of the embryo is recognized by the end of second month or eighth week of intrauterine life. The most observable feature in the fourth week is the formation of the mesodermal somites. These somites are present in the midline on each side of the notochord. These are segmented mesodermal bars. The somites are visible surface elevations which appear between the 20th to 30th day of development. So this period is usually established as the somite period. The age of the embryo is finally identified as the pre-somite period, that is between 15th to 20th day, that is the third week of development, where finally the three germ cell layers are established. Somite period is between the 20th to 30th day of development, that is between the fourth week to eighth week of intrauterine life. Post-somite period is expressed as the crown rump length, that is S is the sitting height, and crown heel length, that is the standing height. Pre-somite period 15th to 20th day that is the third week of development, is therefore related to the formation of primitive streak, notochord, intraembryonic mesoderm, endoderm, and ectoderm. The somite period is 20th to 30th day, that is during the 4th to 5th week of intrauterine life. 42 to 44 somites appear during this period. They are all derived from intraembryonic mesoderm, the second germ cell layer, on the either side of the notochord. Post-somite period is expressed as 5th week onwards, that is crown rump length. CRL is a sitting height measuring from vertex of the skull to the midpoint of the buttocks. Crown heel length or CHL standing height which measured from the vertex of the skull to the heel. So finally, the three germ cell layers which are finally derived from the epiblastic layer. The epiblastic layer via primitive streak forms the endodermal germ cell layer. Next, the epiblastic layer via the primitive streak forms the mesodermal germ cell layer, and the cells which finally remain in the epiblastic layer after the formation of endoderm and mesoderm forms the definitive ectoderm germ cell layer. Derivatives of ectoderm or first germ cell layer starts at the beginning of third week of development. So epiblastic layer, the primitive ectoderm, the cells differentiate into three functional zones. Surface ectoderm, these cells are in the cephalic zone and peripheral margin of the disc and forms a semilunar area. Within the concavity of surface ectoderm is neuroectodermal cells which finally form the central nervous system. Caudally we have pluripotent cells. These pluripotent cells finally form the endoderm and mesoderm germ cell layer. Ectoderm, the germ cell layer, gives rise to the surface ectoderm which finally forms the epidermis of the skin. Neuroectoderm which forms the nervous system. Ectoderm germ cell layer is formed in the embryo. The embryo is broader in the cephalic region and caudally it is narrow. Now coming to the development of central nervous system which starts to appear at the third week of intrauterine life. A slipper shaped plate, thickened plate of ectoderm, which is neuroectoderm cells. These thickened plate of neuroectodermal cells forms neural plate or medullary plate, and it is located in the mid-dorsal region of the embryo in front of the primitive node, which is related to the anterior end of the primitive streak. Appearance of notochord, which appears between the 16th to 22nd day, induces the overlying neuroectodermal cells to thicken to form the neural plate or medullary plate. So neuralization is the important stages related to the neuralization includes neural plate, neural groove, neural folds and fusion, neural crest cells and neural tube, which begins during the late third week of development, that is 18th day, and end by the end of fourth week of intrauterine life by 20th day, is induced by the notochord. Neuralization is finally induced by notochord. Neuralization is formation of neural tube from the neural plate. So neuralization is between 18th to 28th day, starts in the pre-somite period, that is before the 20th day, and almost extends up to the 30th day, that is during the somite period. The notochord induces the overlying neuroectoderm cells to form the neural tube. This starts around the 18th day, forms the neural plate. Neural groove is formed by the 20th day. So under Under the influence of notochord effect that which appears around 16 to 26 day, the overlying neuroectodermal cells, that is neural epithelial cells, thicken to form the neural plate or the medullary plate. The neural plate first appears cranial to the primitive node and dorsal to the notochord. So, neural plate is dorsal to the notochord and intraembryonic mesoderm and also the dorsal aorta. Somite period, which extends between 20th to 30th day. The changes which occur in the neural plate. The lateral edges of the neural plate becomes elevated and they form folds. And these folds are known as neural folds. The depression in the mid region is known as neural groove. The tips of the neural groove is now lined by specialized cells, and these cells are known as neural crest cells, which becomes continuous with the surface ectoderm. Neural groove which extends from the Hensen's node or primitive node up to the buccopharyngeal membrane or procaudal plate. Next, the neural groove grows beyond the buccopharyngeal membrane with the cranial fold or the head fold. Development proceeds. As development proceeds, the neural plate forms, which forms on the 18th day from the neuroectodermal epithelial cells, approaches in the midline and forms neural folds. Later, the neural folds approach each other and fuse to form the neural tube. Neural folds are formed by the 20th day, and neural tube closure of these folds forms a neural tube which occurs around the 22nd day. Fusion of the midline begins in the region of future cervical region at the fourth or the fifth pair of somite period, that is on the 22nd day. Fusion of the neural fluid occurs first in the cervical region. So fusion of the neural tube. So neural plate which is formed on the 18th day. 20th day we have neural folds, the edges and formation of neural tube around 22nd day which occurs during at the region of cervical region. Fusion occurs first in the cervical region, proceeds cranially and caudally. As a result, the neural tube is formed. So neural development starts around 18th day and by 22nd day fusion begins, and it begins in the region of cervical, future cervical region, that is 4th to 5th somite period. The fusion later proceeds cranially and caudally. So various important stages of formation of neural tube. Notochord is an important structure which induces the neuroectodermal cells finally to form the neural tube. Second stage is for the neural formation of neural plate. Third stage is formation of neural groove. The fourth stage is the formation of neural tube, which occurs at the cervical region. Neural tube separated from the surface ectoderm and lies in the midline, dorsal to the notochord cells. Neural crest cells get separated once the neural tube is formed. Neural tube is dorsal to notochord and the dorsal aorta, which is simultaneously appearing on the either side of the notochord. The cephalic end and the caudal end communicate with the amniotic cavity via the anterior or cranial neuropore or and posterior or caudal neuropore. Fourth week of human embryo development, where the fusion of neural tube occurs in the cervical region, that is on the 22nd day. Cranially, we can see the anterior neuropore and posterior neuropore in the caudal region. Neuropores, which are present at the cranial and caudal end, provides nutrition to the neural tube via the amniotic fluid as blood vascular system is yet to be developed. Closure of cranial or anterior neuropore occurs around the 25th day of intrauterine period and it corresponds to 20 somite stage. Closure of caudal or posterior neuropore occurs at the around 20th day which corresponds to 25th somite stage. 22nd day is the 5th somite period. The neural tube closure occurs in the cervical region. Cranial neuropore closes by 25th day or 20th somite stage. Caudal neuropore or posterior neuropore closes by 20th day which corresponds to 25th somite stage. So neuralization therefore is neuroectoderm cells which are derived from epiblastic cells which is formed around 16th day, formation of neural plate on the 18th day, formation of neural groove on the 20th day, neural fusion, that is neural tube is formed which occurs in the region of cervical part, that is 22nd day, and neural tube is completely closed by 20th day. Closure of anterior is on 25th day, closure of posterior neuropore is on the 20th day. So neural tube is closed by 22nd day, that is 4th somite period in the cervical region. Anterior neuropore is closed by 25th day which corresponds to 20th somite period. Posterior neuropore is closed on the 20th day which corresponds to 25th somite stage. Coming to the applied anatomy of the closure of the neuropore. Alpha fetoprotein increases in the maternal blood as neural pores fail to close and it leads to numerous neural tube defects. So neuralization starts the late third week, 18th day, and it ends by fourth week, that is on the 20th day, 18th day to 20th day. Neural plate to neural tube. Neural tube is completely closed by 20th day, and the size of the embryo is around 4.5 millimeters. Coming to the fate of notochord. Notochord induces the formation of neural tube. Once the neural tube is complete, the cells of notochord are fated to die. They are responsible for inducing the neural plate to form the neural tube. Notochord signals the formation of neural tube, in other words. Once the once the neural tube is formed, notochord cells die. So notochord plays a key role in signaling and coordinating the development of neural tube. Coming to the remnants of notochord in adults, it persists as nucleus pulposus